Basic radiopharmacy. Introduction. What is radiopharmacy? It is the manufacturing, production of radioactive medicines, drugs. Here we will present diagnostic radiopharmaceuticals, also known as radiotracers or just simply tracers. So how do we do it? We add a radioactive nuclide to a precursor. A precursor is something that becomes the radiopharmaceutical, the tracer, once the radionuclide has been added. Sometimes only one step is needed, and sometimes many production steps are needed, synthesis steps. Radionuclides can either be produced by an accelerator, and here we see an accelerator. It has an ion source in the middle. It has a vacuum chamber. It has two Ds, which change polarity. It has a magnetic field, and the particles are accelerated between the Ds, which change polarity, plus and minus. And finally, when they've been accelerated, they extract out, and you have a nuclear reaction in the target. To make carbon-11, we irradiate nitrogen-14, and there's a P-alpha reaction. To make F-18, we irradiate enriched water, and it's a P-N reaction. Radioactive nuclides can also come from generators. And a radionuclide generator allowed to separate chemically short-lived radioactive daughter nuclei with good characteristics for medical imaging from long-lived radioactive parent nuclei. Typical techniques used are chromatographic absorption, distillation, or phase separation. This method is in particular applied for the separation of the short-lived technetium-99m, which has a 6 hours half-life, from the long-lived molybdenum-99, which has 2.7 days half-life. Applying the radioactive decay law of the growth of the activity of the daughter nuclide A2 with respect of the initial activity of the mother nucleus A1 can be expressed in terms of the respective decay constants. So you see, molybdenum decays slowly, and technetium is eluted off, and then the radioactivity is built up. A transient equilibrium is a condition reached when the half-life of the parent is approximately 10 times greater than the half-life of the daughter. A classical example is the Moly-99 technetium-99 metastable generator, where the rates of the half-life is 67 to 6 hours, which is 11 to 1. Transient equilibrium is reached in roughly 4 half-lives of the daughter. For technetium-99, the time to equilibrium is 23 hours. Secular equilibrium is a condition reached when the half-life of the parent is many times greater than the half-life of the daughter. Example given more than 100 times greater. This applies for the germanium-68, gallium-68 generator, where the half-life of germanium is 271 days and the half-life of the gallium is 68 minutes. Thus, parent-to-daughter ratio is 5,700. A secular equilibrium is reached roughly in 6 half-lives of the daughter. So this is how a technetium-99m generator looks like. You have the molybdenum-99 on the matrix. You have a vial with sterile saline, and then you apply a vacuum vial, and then the saline is pushed through, and it elutes off the technetium-99m. And here you can see how the molybdenum-99n decays to technetium-99 metastable. And technetium has a very nice property. It decays with the gamma ray on 143 kiloelectron volt. As mentioned, germanium-68 is also another generator. We have the germanium-68, which decays to gallium-68. However, the gallium is eluted with hydrochloric acid and not sterile saline like for the technetium-99. So we're going to show a one-step synthesis of sestamide. So here is a vial with sestamide. This is how the sestamide precursor looks like. We put the vial on the top here on the vacuum, and thus the saline is sucked through the generator. It elutes of the technetium-99m. And once we have the technetium in the vial, we put it in hot water and boil it for 10 minutes. And then the sestamide is done. As radiopharmaceuticals are measured by radioactivity and not by mass, we need a dose calibrator to measure the radioactivity. So here we see the radiopharmaceutical. It's sitting in a well. The well is surrounded by a cylinder which contains a gas over pressure. And when the radioactivity goes through the gas, it creates a voltage. And the voltage is amplified. And then you select which radionuclide it is. You make isotope correction. And then you can see how much radioactivity there is. This is how a commercial dose calibrator can look like. And here you see the well where we put in the radiopharmaceutical. And this is the ionization chamber. And here is the display when you can read out to see how much radioactivity you have. Good manufacturing practice. You need to have it for patient safety and for investigation efficacy. And good manufacturing practice demands controlled and clean hygienic environment, that the manufacturing process is clearly defined. You need written instruction, so-called standard operating procedure, SOP. Operators are trained, records are made, all steps are documented. This is needed for traceability. But why do we need and want radioactive drugs? The gamma radiation gets out of the human body and is measured in detectors. So first we inject the patient with radiopharmaceutical. We wait until it accumulates in the organ of interest. And the gamma rays that are emitted, you can see, they go to the camera and they create an image so the doctor can see how the patient is doing. Single photon emission tomography is when you have single gammas that are emitted and they go through to get measured on the detector. And in order to know from where the gamma comes, you have lead collimators. You can see the black lines. And the collimators do so the camera knows where from the signal comes from. For positron emission tomography, you have an unstable nucleus that decays by emitting a positron. And when the positron combines with the electron, which is its antimatter, it will annihilate and give two gammas going in opposite directions. And because the gammas go in the opposite direction at the same time, we measure coincidence. So when the camera sees two events at the same time, it knows that it's an event and thus records it. If we compare single photon emission computerized tomography versus positron emission tomography, we have single gamma rays passing through the lead septum and getting detected by the camera. So there comes many single events. And in PET, we don't have any collimators, but we detect coincidences. That is when two events occur at the same time in the camera. So how does an image look like? Here we have a sestamibia of a patient, and you can see there's some symmetrical uptake here and here. But this thing looks strange, and that's right, that is a cancer. Here, again, it's a sestamibia image. We have the nice symmetrical uptake in the salivary glands, but this one is suspected. And yes, that's also a cancer. SPECT stands for Single Photon Emission Computed Tomography. If we go for multi-step synthesis, we're going to do the PET radiopharmaceutical fluorine-18 FDG, which stands for fluorodeoxyglucose. So we have the precursor here, and we add the fluoride to it, the fluorine-18. So the fluorine gets to the glucose analog, and then we remove the protective groups. And finally, we have the FDG here, which is a glucose analog. And you can see, this is how glucose looks like, and this is how FDG looks like. So there's an F18 atom instead of a hydroxyl group. And this is an image of a patient we taken with FDG before chemotherapy. You can see there is a lot of bone lesions in the patient. And then we do FDG after chemotherapy. You can see the lesions are gone. So you can follow the therapy using FDG PET. And PET stands for positron emission tomography.
Now we are going to talk about SPECT radiopharmaceuticals, which are the oldest and most widely used type of radiopharmaceutical, introduced in the 50s and greatly expanding in the 1970s when technetium-99 became available. Originally, these were used for planar gamma camera imaging, but increasingly they're being used for three-dimensional imaging with SPECT. The most commonly used radionuclide is technetium-99M, which, as we've already heard, is produced by a generator. Two of the other radionuclides are iodine-123, which is good for labeling small molecules, and indium-111, which is good for labeling peptides and proteins. Some of the advantages of technetium are its availability from an on-site generator right in the hospital. The half-life of 6 hours is a good trade-off between supply, time for imaging, and not giving the patient an excessive radiation dose. The gamma energy is suitable for current gamma cameras. It's a high enough energy that it gets out of the body without too much attenuation or scattering, and yet is easy to detect with the crystal in the camera. There are no alpha or beta emissions, which would increase the radiation dose without giving any more diagnostic information. And the chemistry is not simple, but it can be tamed, and a variety of complexes can be made with technetium-99M. The most common technetium studies are the bone scan using bisphosphonates, mainly for detection of metastases from certain primaries, but there are other uses as well. Myocardial perfusion imaging is an area that is still growing with the two technetium-labeled complexes, hestamib and tetraphosphamide. This is very good for detection of coronary artery disease and risk of future problems. In most departments, these two studies make up well over 50% of the volume of nuclear medicine. Other studies include the emergency lung perfusion and ventilation study for diagnosis of pulmonary embolism, kidney function studies, and sentinel node localization prior to surgery. The form that technetium comes from the generator is pertechnetate, TCO4-, in which the technetium atom is present in the +7 oxidation state. This can be used for thyroid imaging, but in order to make complexes, we need to first of all chemically reduce the technetium to a lower oxidation state, usually with stannous chloride, and this can be performed in what we call a kit procedure in a single vial that contains all the non-radioactive components in pharmaceutical grade form, and the labeling reaction takes place often at room temperature. Here we see an outline of the chemistry. So technetium pertechnetate is reduced by stannous ion to some sort of an intermediate, which may or may not be stabilized by a weak chelating agent. Technetium then forms its complex with the ligand to form the desired product. Usually we want this to be in at least 95% yield. We can see there are two side reactions. There could be hydrolysis to a colloid, which would show up in the liver and spleen on the image. This is a rarely seen impurity, and generally if there's a formulation problem, either pH too high or too much stannous chloride. The more common impurity is free pertechnetate, resulting from oxidation of either the intermediate or final product, and this is seen as activity in the thyroid and stomach. To give an example of a kit preparation, let's go back to hestamib, developed for myocardial perfusion imaging, but also being used for parathyroid adenomas and characterization of some types of tumor. The most commercially successful radiopharmaceutical in history. It came off patent a few years ago, and now there are generic equivalents available. The chemistry looks simple when it's written out here. Reducing agent, reducing technetium, binding to 6 MIB molecules, methoxyisobutyl isonitrile. This requires heat and some time, 10 minutes in a boiling water bath generally, and we end up with this complex where the technetium atom is hidden in the middle with 6 of the lipophilic MIB moieties coordinated around it. The kit formulation contains the MIBI itself as a copper tetrafluoroborate salt, stannous chloride. There's an additional reducing agent, cysteine, which also acts as a weak chelating agent. There's sodium citrate, which is both a buffer and a weak chelating agent. And finally, there's mannitol, an inert filler, just to make it look like there's something in the vial. Once the labeling reaction has taken place, we have to check that the product meets a variety of quality control parameters. A number of them are measured quite easily, as we can see in this table, but we'll talk a little bit more about radiochemical purity testing. Generally, for technetium radiopharmaceuticals, we use thin layer chromatography, which involves placing a spot of the radiopharmaceutical on a strip. In this case, for sestamib, it's aluminium oxide on a plastic backing. The strip is then placed in a chromatography tank with some solvent, in this case ethanol. Solvent migrates up the strip, separates the components, and then we look at the distribution of activity along that strip using a radiochромatogram scanner, phosphor imager, even a gamma camera, or we can cut the strip into two portions and measure the activity in a dose calibrator or a well counter. We then calculate the purity from knowing where the bound and free components will migrate. Here's an example of a scan of a chromatogram with sestamib. Here we can see that 97.5% of the activity has migrated up to the solvent front in sestamib peak, and there are 2.5% impurities remaining at the origin. So, to summarize, SPECT radiopharmaceuticals are the most widely used class, with technetium making up probably 80–85% of all studies. The technetium is obtained on-site from a generator. Similarly, the products are prepared on-site from using kit procedures in GMP-compliant facilities with aseptic conditions. And then there is rapid and simple quality control, which is performed before the product is released for use in patients.
Gallium-68. Presentation overview. We're going to speak about the germanium-gallium generator, the basic principle, how to elute the generator, chemistry, basic principles, synthesis, optimization, validation, hot cells to work with gallium-68, quality, and finish off by the imaging properties of gallium-68. So the generator contains of germanium-68, which has a very long half-life, germanium-68, and the daughter gallium-68. And compared with the technetium generator, which is eluted once per day, sometimes twice, you can elute the germanium-gallium generator several times per day. Just let it wait a few hours between elutions so it goes back into higher activity. When you elute of gallium-68, you do it with hydrochloric acid and not saline. And there are different methods to get it. The first and simplest method is just called fractionation. Then you have anion exchange solid phase extraction, also a chromatographic method, and cation exchange solid phase extraction, which is also a chromatographic method. So when we elute the generator, we can see there's very little activity in the first half a milliliter, but then there's more in the second half milliliter. And once we have eluted of the third one, it's even more, and then it decreases and goes down. So rather than taking the whole volume, we start to elute after we have discarded the first half milliliter, and then we collect the three fractions here in the middle, and then we discard the rest. So we just collect 1.5 milliliter. So only this portion is used. And the idea behind it is that you will have the activity more concentrated, and you will have 70% of the activity in 1.5 mil rather than having it in 7.5 milliliter. So this is called fractionation. Anion exchange solid phase extraction. You load the gallium onto the column in 0.1 molar hydrochloric acid, This is a strong ion exchange column, and the gallium gets stuck on the column and everything else goes to the waste. Then you clean the anion exchange column with acetone and with diluted hydrochloric acid, and this keeps still the gallium on the column, and you have impurities like a breakthrough of germanium going to the waste. And finally, you elute off the gallium with acetone and a small amount of hydrochloric acid. So your final product, the gallium, comes on in acetone and hydrochloric acid. Here you need to remove the acetone before the synthesis so it doesn't end up in the ready radiopharmaceutical. In the cation exchange solid phase extraction, you have the gallium-68 in 0.1 molar hydrochloric acid. This is how it comes off from the generator. Then you mix it with concentrated hydrochloric acid. And once you have the gallium in roughly 5.5 molar hydrochloric acid, you put it on a strong cation exchange resin, and the gallium gets stuck on the column and everything else goes to the waste. And then you take water, put it through the strong cation exchange resin, and you have the final product, gallium in hydrochloric acid, because the water is it doesn't contain any hydrochloric acid, but the residual hydrochloric acid on the column will come off, so we'll have the gallium in a slightly acidic solution. And another version of the strong cation exchange method is the called sodium chloride method. So you take the gallium from the generator in 0.1 molar hydrochloric acid, you put it on the strong cation exchange resin, the gallium gets stuck on the resin, everything else goes to the waste. And then you take sodium chloride with a small amount of hydrochloric acid, and you elute off the gallium. And the advantage here is that you have it in a very small volume and that the hydrochloric acid is not so strong. Here is just an overview of different germanium-68 gallium generators, and they look pretty much the same. Some are square, some are circular, and all of them are eluted with hydrochloric acid, and normally it's 0.1 molar hydrochloric acid. Chemistry. We're going to discuss a little bit the stability of the chelators, open versus closed, about pH control, and about metal environment. Some properties of gallium. Gallium behaves like iron in vivo and binds to transferrin in the blood plasma. Gallium is mainly excreted through the kidneys. Germanium-68 has 271 days half-life, and gallium-68 has 68 minutes half-life. There are different kinds of chelators. We have the open one, which you can see here, the DTPA and the EDTA, and the closed one, like NOTA and DOTA. And generally, the closed chelators are more stable than the open ones. And this is the stability of some lanthanides in the chelators. And this is the size of gallium. However, due to the coordination chemistry, gallium is very stable in DOTA and NOTA. And you can see that gallium has 6 coordination chemistry. It binds to 1, 2, 3, 4, 5, 6 atoms in the NOTA, and the same in the DOTA. 1, 2, 3, 4, 5, 6. So gallium has the 6 coordination chemistry. It's very important when you have making gallium labeling to have the pH control. And you can see that the amount of free gallium decreases when the pH increases. And normally we do the labeling between 3.5 and 4.5. If the pH goes too high, you're going to have gallium hydroxide, and this precipitates, and thus no chemistry is possible. Once you have labeled your DOTA or no gallium, and it's much more stable, and you can see that it's very stable over a range of pH pretty much 0 to 9. And the point with this is when you make the radio labeling at the pH which is very low, you maybe want to adjust the pH to a higher pH to make it the final radiopharmaceutical injectable. So once the gallium is in DOTA, you can adjust the pH to a higher one than the labeling conditions. Metal-free environment and conditions. No metals must be in contact with any of the reagents and chemicals for the manufacturing of gallium-68 radiopharmaceuticals, with the exception of needles for the fast transfers of example given the precursor or the finished gallium-68 radiopharmaceutical. You should avoid glass containers. Example given the hydrochloric acid used for dilution of the generator should be stored in a plastic container rather than in glass, as the acid will etch away metal ions from the glass after continuously. Metal-free or low metal content chemicals should be used. When you're going to make synthesis and method validation, it's good to start with different amounts of gallium-68 and different amounts of your DOTA precursor. And if you should normally have a reaction time for at pH 4.3 at 90 degrees for 10 minutes. If you have too short reaction time and too low temperature, it may potentially give a free and unreactive gallium-68. However, too long reaction time and elevated temperature may produce side products from thermal decomposition. So here is just a matrix which starts around pH 4.3 and 90 degrees, and you react from 8 to 12 minutes. So you start at pH 4 at 80 degrees for 8 minutes. You make pH 4.6, 80 degrees, 8 minutes. Then pH 4.3 at 100 degrees for 12 minutes. And for pH 4.6, 100 degrees and 12 minutes. And then finally 3 times pH 4.3 at 90 degrees for 10 minutes. And once you've tried this matrix, you will know which reaction conditions are best suited for your compound. There are different hot cells for gallium. Here is one example, here is another, and here's a third one. And compared with technetium, which doesn't need much shielding because it is a soft gamma emitter, gallium generally needs 5 centimeters of lead protection due to the 511 keV gammas. And this is how a synthesis of DOTA-NOx looks like. So here is the peptide, and here is the closed chelator, the DOTA, and here is the gallium. Once it's been mixed and heated, the gallium gets trapped in the chelator, and then it remains very, very stable. If we compare gallium-68 versus fluorine-18 in imaging, we can see that the gallium has much higher endpoint energy than the fluoride. Thus, the positron range is much longer for gallium, especially in soft tissues, as compared with F18. So the image resolution is better for fluorine-18 than for gallium-68 because fluorine has a lower positron energy than gallium. And here we can compare the fluorine-18A and B with gallium-C and D. And you can see that in this phantom you have a very nice resolution with F18, but with gallium-68 the image can get very blurred. Quality control, or just QC. For the chemical and radiochemical purity, high-pressure liquid chromatography, HPLC, is normally used for validation, and then instantaneous thin-layer chromatography, ITLC, is used for routing control. pH can be measured by paper strips, and the radionuclidic purity should be tested at least once for each new generators. For microbiological testing of the finished radiopharmaceutical, you test the bioburden during validation and then roughly every 20th synthesis. The same for sterility, every time during validation and then every 20th synthesis. And also the same for the endotoxins, the AL test, every time during validation and then every 20th synthesis.
This presentation teaches the production and quality control of the workhorse tracer of PET. This is 2-fluorine-18 fluorodesoxyglucose. The content of the presentation is as follows. First, a brief explanation is presented on the physiological uptake mechanism of FDG in tissue and why uptake is increased in tumors. Then we will focus on the production of the radionuclide fluorine-18, which is produced by cyclotron irradiation. Properties and principles of the cyclotron will be discussed. The next point is the workup of cyclotron-produced fluorine-18 fluoride to make it suitable for the radiosynthesis of FDG. The radiosynthesis will be discussed both theoretical and practical aspect. Finally, attention will be paid to the quality control of FDG in combination with the release criteria as formulated in the World Health Organization Pharmacopoeia. By performing an FDG scan, the aim is to measure glucose consumption in tissue. Glucose consumption is increased in tissues with increased energy metabolism, such as tumors, inflammatory tissue, brain, and the heart. These tissues display therefore high FDG radioactivity uptake. The uptake of FDG in the cell is similar to normal glucose, as the first steps are concerned. Both sugar molecules are actively taken up by the GLUT1 transporter. In the cell, the sugar molecules enter the glycolysis pathway. Unlike glucose, which is completely metabolized through glycolysis, Krebs cycle, and oxidative phosphorylation, FDG is phosphorylated to its 6-phosphate analog by the enzyme hexokinase. However, FDG 6-phosphate is not further metabolized, but irreversibly trapped in the cell. Tumor cells that have increased expression of the GLUT1 transporter and hexokinase therefore show increased radioactivity uptake, resulting in hot lesions on the PET scan. For the production of FDG, the radionuclide has to be prepared by a cyclotron. Modern cyclotrons accelerate H- particles to a high energy. In the ion source, at the center of the cyclotron, hydrogen gas is broken down into a proton and an H-. H- is pulled out and accelerated by a high-frequency alternating voltage, and its motion is bent into a curved trajectory by a perpendicular magnetic field. The H- speeds up each time it crosses the electrical field. Because of the acceleration, the kinetic energy is increased gradually, and the perpendicular magnetic field causes H- to travel in a spiraling horizontal path. After several hundreds of circles, the energy of the particle has reached a value of 17 to 18 mega electron volt. Finally, at the edge of the cyclotron, 2 electrons are stripped from the H- when it goes through a carbon foil. The left image displays a schematic view of the cyclotron. The right image is a photo of the inner part of the cyclotron. The resulting H+ particle is directed to the fluorine-18 target filled with O15 water. The protons produced by the cyclotron collide with the O18 nuclei to form fluorine-18. After irradiation, the resulting radionuclides are released from the target to the hot cell in the laboratory. Several hundreds of gigabecquerel fluorine-18 can be produced. When the irradiated O18 water containing the produced fluorine-18 has arrived in the hot cell, the water must be removed from the fluorine-18. It is very important to obtain dry fluorine-18 fluoride, because the presence of even small amounts of water will result in solvation of the fluoride ions, which then are chemically not reactive. Fluorine-18 fluoride is trapped on an anion exchange cartridge and subsequently eluted with a solution of potassium carbonate. Water is removed by adding dry acetonitrile, followed by azeotropic distillation. Azeotropic means that the resulting boiling point of the acetonitrile water mixture is lower than the separate solvents. The distillation is carried out several times depending on the exact distillation condition. even one distillation could be sufficient. Because the resulting potassium fluoride salt is insoluble in acetonitrile, a phase transfer catalyst is added before the distillation process. The most often used phase transfer catalyst is cryptofix, but also tetrabutyl hydrogen carbonate is often used. The methods are presented in the next two slides. Cryptofix is an amino polyether that perfectly complexes the potassium ion. This apolar complex acts as a counterion of fluorinating fluoride that is attached as a naked reactive nucleophile to the positively charged complex. The potassium cryptofix fluoride complex is well soluble in acetonitrile, which is the solvent of choice for the radiochemical synthesis of FDG. The advantage of the use of cryptofix is that the compounds are easily to handle and crystalline. Disadvantage is that cryptofix is toxic and that cryptofix can potentially be present in the final FDG solution. Therefore, careful quality control on cryptofix is required. In addition, the reaction conditions of the fluorination are always basic because of the presence of carbonate ions. This may result in formation of side products. As alternative phase transfer catalyst is tetrabutyl ammonium hydrogen carbonate used. This positively charged cationic compound acts as counterion of fluorinating fluoride, while because of the four butyl groups, its apolar nature enables its easy dissolving properties in acetonitrile. The advantage of using tetrabutyl ammonium hydrogen carbonate is that neutral fluorination conditions can be applied with theoretically less side products. Moreover, it can be easily removed from the final product by passing through a cation exchange cartridge. The compound can be purchased as a solution in water. Once the fluoride is dried and present as a reactive fluoride in acetonitrile, it is mixed with the mannose triflate precursor, here depicted on the left side of the scheme. The sugar molecules contains four acetyl groups that protect the hydroxy groups from reaction with fluorinating fluoride. The leaving group triflate at the 2 position of the sugar is substituted by the nucleophile fluorinating fluoride following the SN2 nucleophilic substitution reaction mechanism. In this reaction, triflate is substituted by a backwards attack of fluoride, resulting in inversion of the stereochemical configuration. In this way, the sugar conformation mannose in the precursor is changed to glucose in the final product. An evaporation step afterwards can be used to remove acetonitrile. The next step is removal of the 4 acetyl protective groups by basic hydrolysis at room temperature. If the temperature exceeds 40 degrees Celsius, racemization can occur, resulting in the formation of fluorodesoxymannose. The final product is purified by solid phase extraction method. The nucleophilic substitution between mannose triflate precursor and fluorinating fluoride requires heating. Temperatures between 80 and 110 degrees are usually applied, with typical reaction times of 5 to 10 minutes. The hydrolysis of the acetyl protective groups are performed under basic conditions, usually by adding a solution of sodium hydroxide. As already said before, Nowadays, several synthesis modules, as shown on the slide, for routine production of FDG are commercially available. These modules can be based on disposable The radiochemical yields for FDG are very high, mainly because a large amount of precursor is being used. The yields vary between the several synthesis modules and are between 50 and 80% corrected for radioactive decay. The total synthesis time, including the purification steps, is usually slightly faster than half an hour. After the radiosynthesis, quality control is being performed in order to assess if the radiopharmaceutical meets the criteria as described in the pharmacopoeia. If FDG complies with the requirements, the qualified person will release FDG for human use. As the workhorse tracer of PET can be produced in large amounts, up to several hundreds of gigabecquerel, FDG can easily be distributed to PET sites without a cyclotron, located within 2 or 3 hours from the production site. The release criteria for FDG are described in several pharmacopoeias, including the International Pharmacopoeia of the World Health Organization. The criteria are quite comparable amongst the different pharmacopoeias. Besides the generic methods that apply for all radiopharmaceuticals, there are specific requirements for FDG. For most PET radiopharmaceuticals, including FDG, the radionuclide purity must be higher than 99%, as determined by gamma spectroscopy. Radiochemical purity must be higher than 95%. This means that fluorine-18 fluoride must be lower than 5%. Of the 95% fluorine-18 product, more than 90% should be FDG, where maximally 10% may be FDM, as a result of racemization during the radiosynthesis. The maximum allowable cold FDG present in the patient dose should not exceed 10 mg. In addition, there are requirements for the presence of phase transfer catalyst in the final product, as both are toxic.
This part of the webinar deals with GMP and radiopharmaceuticals. GMP is an acronym of Good Manufacturing Practice, and it may be defined as that part of the quality assurance which ensures that the products are consistently produced and controlled to the quality standards appropriate to their intended use. GMP is a system of principles and rules created with the aim to protect the patient, ensuring that the medicinal products are prepared with appropriate quality, safety, and efficacy. Please remember that radiopharmaceuticals are medicinal products. Indeed, GMP are legally binding for industrial manufacturers, and in several countries they are legally binding also for radiopharmaceuticals prepared in hospital radiopharmacies. In case they are not legally binding, at least a suitable quality assurance system needs to be in place. GMP ensure traceability of the preparation operations. If you want to get familiar with GMP principles and aspects, there are several sources to gather information. So, you may download GMP guidelines from the website of the World Health Organization, WHO, or the website of the European Commission, again from the website of Pharmaceutical Inspection Convention Scheme or the Food and Drug Administration. As said before, GMP is a system of principles and rules created with the aim to protect the patient, and their contents are very similar, as you may see here from the comparison between European Union and World Health Organization GMP. Moreover, they cover really most of the topic included in the preparation of a radiopharmaceutical, with personnel, premises and equipment, documentation, production, quality control, complaints and product recall, self-inspection. Also, GMP guidelines include parts which are specifically dedicated to the preparation of radiopharmaceuticals, such as the Annex 3 of WHO guidelines and the European Union GMP guidelines, or the chapters dedicated to the current good manufacturing practice in the preparation of pet drug released by the FDA. But what does it mean in practice to comply with GMP? Personnel should be appropriately qualified and sufficiently trained. Responsibilities should be clearly identified, and responsible for the preparation and quality control of radiopharmaceuticals should be independent to avoid possible conflict of interest. It is very important that every single action and operations are documented to ensure traceability. And a very important part of every quality assurance system are the so-called Standard Operating Procedure, or SOP, which include instruction for the preparation, quality control, quality assurance, as well as specification for. Standard operating procedures are aimed to provide specific and detailed instructions on, for instance, how to use an instrument, or how to collect and record data, or to set limits and acceptance criteria for the quality control. Here you may see a typical index of an SOP, but of course other format may be used. GMP defines characteristics and requisites for premises and equipment, which should be designed so as to minimize contamination and to allow for effective cleaning. This is very important if we remember and we consider that most of the radiopharmaceuticals are in the form of sterile injectable solutions, so microbial contamination has to be kept at a minimum extent. Under this point of view, a very important role is played by the heating, ventilation and air conditioning system, or HVAC, which has to ensure proper air cleanliness. Also, we have to consider that operator safety requires negative air pressure inside the preparation lab, while the product protection requires positive air pressure inside the preparation lab. So pressure regimes should be designed so as to guarantee both product and operators' safety. But even more important is that operating personnel, so we are the major source of microbial contamination. So behavior rules are very important. GMP defines standards on how to handle starting materials in order to be sure that they are of the proper quality and that they are properly labeled, accepted, stored and controlled. GMP states that equipment should be qualified prior to their use and preparation processes should be validated. A very critical point due to the radioactive nature of radiopharmaceuticals is that radiopharmaceuticals need to be controlled before their release. Even if we are working correctly, following the standard operating procedures and quality assurance principles, we may never rule out the possibility of a failure or a problem in general. So it is very important to define procedures on how to handle deviations, analytical results out of specifications, and major changes to instrumentation or to analytical method or to preparation steps. All of this operation should be documented, of course, and potential risk considered. Risk assessment is a useful tool that should be used to help operators and responsibles to take decisions. Risk assessment is even more important in case of radiopharmaceutical, and it is indeed considered as an integral part of all the activities related to the preparation of radiopharmaceuticals, including validation and qualification. An insufficient risk evaluation may increase the probability to failure or to have problems. To conclude this presentation, we can say that the application of GMP to the preparation of radiopharmaceuticals is feasible, but it is not always trivial. Indeed, we have to consider that GMP were originally designed for the preparation of normal, non-radioactive drugs by the industrial manufacturers, while radiopharmaceuticals is a peculiar class of pharmaceutical due to the radioactive nature of the radionuclides. For instance, we may consider the following points. Half-life of the radionuclides is often short or even very short. For example, the half-life of carbon-11 is only 20 minutes, so the operations need to be carried out quickly. Also, the short half-life does not allow to perform all the quality control tests before the release. For example, sterility test and radionuclidic purity test need to be performed after days. Full GMP application requires significant human and economic resources to comply. And often hospital radiopharmacies are too small for full GMP application, so they have tried to adapt GMP to their size and to their resources. Often, radiopharmaceuticals for diagnostic purposes do not have any pharmacodynamic activity, so low or not at all toxicity, which should reduce in part their problems. Again, most of diagnostic radiopharmaceuticals have a very short shelf life, from minutes to hours. The batch size of a typical radiopharmaceutical preparation is very small. Often it is a single vial or a few vials at worst. Also, the labeling of the vials of the primary and the secondary container needs to be adapted to the radioactive nature of radiopharmaceuticals. So, in conclusion, GMP needs to be adapted and suitably designed in case of preparation of radiopharmaceutical. And we never have to forget that we have to consider radiation protection of the operators. And here you may find useful web reference. Thank you for your attention.
Summary. In this webinar, the following topics have been covered. How radionuclides are manufactured, either by cyclotron, such as fluorine-18 and carbon-11, or by a generator, such as technetium-99m and gallium-68. The two different types of diagnostic radionuclides, single photon emitters, such as technetium-99m and indium-111, and positron emitters, such as fluorine-18 and carbon-11. How radiopharmaceuticals are manufactured, either by a one-step synthesis, such as sestamib, or by a multi-step synthesis, such as fluorine-18 FDG. How the amount of the radiopharmaceutical is measured, in a dose calibrator. How radiopharmaceuticals are manufactured, using good manufacturing practice, GMP, and following standard operation procedures, SOPs, how the quality control of radiopharmaceuticals is performed, chemical, radiochemical, radionuclidic, and microbiological purity, what diagnostic radiopharmaceuticals are used for, oncology, example given using fluorine-18 FTG, cardiology, example given using technetium-99 sestamibi. This field is called nuclear medicine. Thank you for the attention. In alphabetical order, Jim Ballinger, Philip Elsinga, Jacek Koziorowski, and Sergio Todde.